Okay, hello. So my name is Husen Umer. Right, so I will try to. So let me just give a short background on this topic. That is a proteogenomics study, basically to analyze the reanalyze some proteomics data sets with the goal of identifying or improving the current reference proteome database. So I usually feel I'm a stranger in the room when there's some computational proteomics people around, because I'm coming from the genomics background, and my hobby is to look into genomics data or like advances, and then see how we can translate this into proteomics issues. Let me just I think you are all aware of these long read technologies nowadays. So basically we get longer reads, and that means actually we can improve the genome assembly. Unfortunately, there was this pangenome consortium, so they resequenced a large number of the previously sequenced samples from the 1000 Genomes project that were before sequenced with short read. And now with this resequencing, so we get better reference genomes. So there were already some papers published from this consortium, and so about 96 samples are publicly available with the entire genome. So there are two good things with these new genomes. So first, the genome is improved so that in the repeat elements and all the complex regions, it has a better alignment now. And the second is we get better calls for variants, genomic variants. So these are like population variants, because these are like normal samples. Okay, so the reason for this, that we want to look into this, is because I personally don't trust that, maybe that's too harsh to say, but the reference gene proteome is usually not complete as like the reference genome, right? So there should be some improvements to cover, which I think it's not really one database that should be. That's the idea. The pangenome usually have variants at different sample level or population level, and I think that the same thing should happen at the proteomics database level. So anyway, so to this end, so the goal is basically to relook into the proteome, like the protein reference database, and see if we get new peptides based on these pangenomes. So what we do is we got these annotations from these 96 samples, and then we have this current T2T, like telomere to telomere reference genome right now, which we should be using more often, but still. So we make like one large database from this. So we got all the proteins from this improved genomes, and then we add the canonical like ensemble or UniProt database. And then this provides us with this proteogenomics database that looks huge, or it feels huge, because if you look at this figure on the left, so the number of proteins is like 2.5-fold of the current number of proteins in the UniProt database. However, if you look deeper, so if you digest these proteins into tryptopeptides, so then you only add 15% extra tryptopeptides to the database. So the database size is not actually increasing if you think about search engine approach, but it looks like it's huge, but it's not huge. So anyway, so that is what we have. So in the far left, okay, I'm talking about my side. Like I'm really bad at directions. So we have some 123,000 GCA peptides. So these peptides are only found in the pangenome or reference genomes. And then we have a smaller number of canonical peptides that are not found in any of these samples. So these are still like theoretical peptides, right? Yes, but that is the content of our database that we will search. And then to search this data, so we use this CompMS pipeline that was recently published. And it's quite nice because it allows you to use multiple search engines, and then it takes like SDRF as input so that you can annotate your data sets. And then at the end, you get MZ-tab output. So anyway, so we select two big data sets from the PRIDE or Protein Exchange, and this contained more than 90 million like spectra and covering more than 30 normal human tissues. Right. So the result of this basically is, so we have the canonical, if you look at the canonical peptides in the first violin plot, so the error like PEP, probability error, whatever, is very similar to the GCA peptides, and it's very different compared to the decoy and the other the decoy peptides, basically. So this is already an indication that the GCA peptide probability is quite similar to the canonical one, so we are not doing really bad. But still, if you look at the distribution, it's a bit actually larger, so we have more error in the GCA peptides. So GCA means the novel peptides, okay? So that means we have more work to do to validate or to make sure the peptides are actually true peptide identifications. So this was only from one of the data sets, but similar to the other data set also. So for this reason, to make sure that the peptides are actually correct, so then we do multiple step like validation, and that includes first removing all peptides that are present in other canonical databases, and then doing alignment to like blasting across databases, and then we use two different approaches. So first, spectrum AI. So that's basically validating places where you have single amino acid differences, and then it looks at the B or Y ions, and then to make sure that that position has enough support. And then MS2PIP and also DeepLC basically looks at the retention time of the predictor on your actual peptides. I think you know better than me in this, so I don't spend time. And then finally, we get our list of peptides, and these are the number of peptides or spectra, actually. And then how many we remove at each step. So as you can see, with this validation, we remove about 40% of the total identifications, but we are still left with a good number of GCA peptides, and that is basically 4,991 GCA unique peptides. I think we have like 80,000 or more spectra supporting this. And more than 50% of these have never been seen before because they were not included maybe in the databases where people were searching the mass spec data. And importantly, so more than 90% of the GCA peptides have single amino acid variants. And the reason for this is because we are looking at sample-specific variant, like variation. So it's kind of more related to the population variants. Is my time running fast? Okay, good. So if you look at the like where these peptides are located, so then you map them to the genes, and then because at the end we are interested in what genes have more of these peptides, right? So the majority of genes have only one variant peptide, but there are actually a large number of genes that have multiple variant peptides, and so these are only genes that have more than 5 variant peptides. So basically, they have 5 different places of the gene that is mutated, or and then I think that's a complex figure. Maybe I can skip it. But the important thing here is because for those who have done mutation calling from genomic studies, usually, so the larger the gene is, the more mutations you identify. So you want to make sure that's not the case here. So there's no correlation between the length of the protein and the number of mutations, except this TTN, which is a very problematic protein or gene usually. Right. And that's an example of what I said. So for example, if you take this gene or protein, so there are 8 different positions of protein that are mutated, and these mutations or single amino acid variants are actually found in different samples or different populations. So then we have a large number of spectra supporting each of these different variants across the data sets. And they are also identified in different tissues. So it's quite, I think, reliable. I'll skip this. And so now if we take this problem back to where we started, the population genomics, because that's the idea with 1,000 Genomes project usually. So if you look at the specificity of these variant peptides, so in the top, we have the genes. So how many of these variant genes are found in each population? So there is like 435 genes that are identified in all populations. So that means 2 things, because these are genes, so you could have different places of the gene. But if you look at the figure below, so the panel below, So then we have the African and the South American population that have the largest number of population-specific variant peptides, and that is because we have the large number of samples in these two populations. So this means that we need to actually generate database from more and more pangenome or sample-specific genomes, and then search them so that we can cover the full spectrum of population-specific protein variants, so that when you search, you have a full coverage of what you are looking for. And so the third bar chart, so this shows that we have, I don't know how many, but a large number of peptides that are found to be like the variant peptide that's identified in all populations. And I think this means that these are definitely should be the canonical peptides, and you should replace them in the reference database. Just put these ones in instead. Yeah, with this, I think, yeah, you have this. And I would just like to thank my collaborators, especially Yasset and Dongquang, for basically doing the major job. And this is available in bioRxiv. And yeah, thank you for listening.